In this transpiration lab, we're going to measure the rate of water loss by these plants by their transpiration through their stomata. On the undersides of these leaves, we know that there's little openings by which, little pores by which water transpires through. And that evaporation through those pores pulls water by bulk flow from the roots all the way to the leaves. And so what we're going to do is we're going to measure this rate of transpiration by measuring the water loss or the loss of mass over time by putting these plants into different environmental conditions. Okay? And so in order to do that, we're going to have to bag these plants. Now these plants are coleus, and coleus are shade growing plants, and we've grown them in our lab for this purpose. Okay, and I tried to pick plants that had about the same colors. Now if you notice, this one is a little bit lighter. Now if you notice, being a little lighter, does this photosynthesize more? So we're going to take this one out, but the rest of the remaining plants have about the same color, and again, we're guessing here a little bit, have about the same amount of surface area of leaves. They're about the same size. So we're going to use 4 of these plants. Okay, we're trying to measure the rate of transpiration under 3 conditions. Those conditions, of course, will be 24-hour darkness, we'll do under wind, and we'll do the last one under 24-hour light. And so essentially those are our variables that we're going to test in order to see which of those 3 variables affects transpiration the greatest or the least. And keep in mind, since transpiration is regulated by our stomata, okay, those guard cells that open and close, this lab is really about measuring how those guard cells work in these what we call C3 plants. Okay, any case, now if we're going to measure the rate of water loss, we're going to make sure, we have to make sure that the water doesn't evaporate, okay, from the top of the plant. So we're going to have to bag these. Okay, so I've got 4 plants, one being control. That control is going to be under 12-hour light. Now we need to bag the plants. Okay, and again, the idea here is to make sure that there's no evaporation, okay, that occurs here. We're trying to make sure that all evaporation, or at least in this case, transpiration, occurs from the top. So just taking a very simple 1-gallon bag, okay, and I'm just going to cut the top off because I really don't need the top. And also, I did make sure that I really saturated every one of the plants before I started, meaning I really watered them really good. So they're not starting out in a dry or flaccid condition. They're all very turgid. So I'm putting the plant in a bag. Okay, and you can see what I'm doing here. Now, what I'm going to do is take a rubber band, and I'm going to gently try to take this extra plastic, kind of stuff it in plant, and I might lose some lower leaves, but that's okay. Okay, so I'm just basically taking this and I'm making sure that all this plastic here is gently pushed into the plant. Again, I do not want to have any of the soil exposed to the air. I don't want any evaporation. Any loss of water would be just due to, of course, the water through the leaves pores, the stomata here. So now I'm just going to put a rubber band gently. I don't want to break the stalk. Okay, that's important. So I'm just going to put this rubber band lightly. I'm going to twist once. Okay. And go around the end. And we want to be careful not to hurt our friends. And that, I think, is going to be tight enough to do what we need to do. And we'll do that for the other 3. So again, the whole idea is to just make sure there's no evaporation. And we'll do that 3 more times. This being my control, or one of the tests, it doesn't matter. So we'll do that 3 more times. So we have our 4 bagged plants. Okay, and now we just need to mass them out. All right, let's label our plants. This is going to be the control. And let's go mass out our control, which will be under 12-hour light. Okay. And then let's go measure our other plants that will be under different variables, different conditions. And so we've got our 24-hour light. Okay. And then we're going to label this one the wind. Hour light with wind. And then this one. This is going to be 24-hour darkness. And now we need to put them in their different conditions.